Thank you, Todd. And yeah, I have a similar story for Oxford Nanopore. So we were one of the first groups that got the demo instruments in 2014. And the question was, does it work? And the answer was no. And so we stuck with it for a very long time, but similar experience. The first time we put a genome together was Klebsiella pneumoniae. Took us 5 flow cells. It had like 30x coverage, and it was junk. So we've been battling this for a very long time. But I'm pleased to say we're past that idea of the technological, can I do this? And we're now into how do we answer biological questions with this? But as we keep going forward, and I'm going to wander. I don't like standing behind a podium, and I've got a mic. So what we started to see here is as we've gone through these different steps, now we're at the point where we can take pretty much any bacteria, sequence it, many different bacteria in a single flow cell, and go through and do all of these different things, and we can generate circularized chromosomal assemblies, circularized plasmid assemblies. But we've also found that as we've kind of gotten to this technological place where we can close these genomes and create a consensus assembly, and I put that in big air quotes, we're starting to see that we've run into how do we differentiate biological reality and variability from technical artifacts and things of that sort. And so that's really what I want to talk about today is what are these challenges that we see? Where are we today? And I use this as somewhat of a call of action for you all to think more about the biology piece, and also we need people answering these questions and developing tools, because I will say straight off, the existing tools do not address these problems. They kind of just ignore them. And so to start out with, I'm sure this is an underestimate, but biology is not simple. If it was simple, we wouldn't be here. We wouldn't be doing this, right? It would have already been solved. We'd be past it. But in that same situation, bacteria in particular use a lot of variability to kind of guard themselves against selective pressures, things like immune response, antibiotics, all of these different pieces, and having heterogeneity in their genes, their genomes for structural variation, and their phenotypic expressions allows them to guard against these large selective events. And so we often see that they have developed many mechanisms of variability to enable them to be more recalcitrant towards these types of challenges. And so here are just a subset of them. And these are the ones that we see the most often, but there are all sorts of different variations. And so you see things like phase variation, which is where you'll have a structure that will actually change the genetic structure to regulate specific genes, and it is an expression change. And so this can be slip strand mispairing, where we actually have a situation where the distance between a gene and its promoter will change, allowing it to either be turned on or turned off. And these are often in some sort of stoichiometric ratio inside of the colony that will be reproducible. And so how do you represent in a consensus genome something that inside of a single colony of bacteria is inherently different? And the answer is, I don't know yet. And I think that's the thing that we need to start to think about as a community, is really understanding how we reflect these things. And it's not just phase variation. We also have things like antigenic variation, where you'll actually have genes that will recombine in multiple ways to create multiple different antigenic surface structures on the bacteria as a way of avoiding the immune system. And essentially the idea there for the bacteria is once the immune system knows it's there, it changes the surface antigens, and all of a sudden it looks like it's hidden again, and then the immune system has to find it again. And it allows it to stick around for much longer, and it basically plays the adaptive immunity hide-and-seek game. We also have things like genomic and pathogenicity islands, which are also similar, but these are much larger, to things like integrons and transposable elements, which often carry genes that enable things like resistance to antibiotics, to metals, all sorts of other pieces, virulence factors that are useful for the bacteria to have, but often come at a fitness disadvantage. So by having that DNA, they end up having to work harder to do the same thing as others. And so you'll see that part of the population will pick these genes up. It will do things like antibiotic sequestration, and then the other part of the population takes advantage, and is kind of a cheater cell, in that it will take advantage of one part of the population working harder, and it hangs around and doesn't have that. And so you have this variability in even that same colony and that same population. And then we have things like prophages and CRISPR-Cas systems, which are where these viruses either in certain parts of the genome, sometimes they'll excise themselves, or the CRISPR-Cas, which is essentially the bacterial immune system, right? It's the idea of that adaptive immunity in bacteria. It will pick up parts of the virus, it sticks it in its genome, and then when it sees that same signal in the future, it chops the virus up really quickly to be able to act as immunity and prevent continued infection by these viruses. All of these different things can represent heterogeneity within a single, quote-unquote, clonal population. And as an example of this is a bit of work that I did in my lab a few years ago with Steve Norris, who's at UT, looking at Treponema pallidum. And Treponema pallidum is an organism that up until very recently had to be grown in rabbit feet because it was a commensal human organism, and that was the only thing that we could really use as a model. So Steve and a few other groups found a minimal set of media to be able to grow this organism in culture, which opens up all sorts of abilities to do more detailed work and study of the pathogen. But what was interesting is in developing this culture, he developed a method where they would inoculate this specific pathogen down to about 0.5 cells per well, which meant that about half the wells didn't grow anything, and half the wells grew, allowing us to really have confidence that in each one of those wells that grew, it was essentially one organism, one cell, that started that process. And by going through, we would sequence a number of them, and you can see here's a linear representation of the circular chromosome for the reference, so this was our inocula, and here are 3 different replicates of that same initial inocula. And as they would go through, what we would see is that there were reproducibly multiple genetic structures, things like insertions and deletions, single nucleotide polymorphisms, repeat structures that would establish, and then you'd go and if you looked at the multiple sequence alignment, you would actually see that there would be a ratio reproducibly of these different bases at that position. So this is a reproducible way that this bacteria regulates itself within a community. And when you generate a consensus, it doesn't reflect the heterogeneity of what's going on in that specific organism. So technically and technologically, we're able to generate this consensus, but then what does this consensus mean? I'm still working on that. Shelby, one of my PhD students, is in the weeds on this right now. She can tell you all the different ways we see this, but I think we need to get past kind of this idea of technological consensus and start to reflect the biological variability that is here, these low frequency variants, how these structures let these organisms adapt and start to get into these pieces. Another really great strong example of this is a paper that was published a few years ago in PNAS, and this is looking at a Staph aureus genome from a clinical isolate that was observed that when you would grow this organism up, it would create these small colony variants. And small colony variants are known to grow slower and essentially allow the organism to kind of outlast antibiotics and the immune challenge, and it makes them much harder to treat and clear. And what they found is if you took a wild type cell and you plated it, about 20% of the organisms would be small colony variants. If you took one of those small colony variants and you plated it, about 20% would be wild type. And so regardless of what was happening, they could not get pure cultures of one and the other, and they found that this was actually because of this inducible variation that was going through based on a mobile genetic element, and it would go through and it would actually turn about a third of the genome and flip it over, causing massive structural change. And you can see here's the wild type and the normal variant here. Here's the small colony variant, and it changes the regulation pretty substantially. These small colony variants actually had reduced virulence, reduced growth dynamics, and made this small colony structure. But again, it's stoichiometric. So it's always phase switching at some portion in the population. And in our traditional short read assemblies, they would just fracture at that point. We would never be able to recombine that. It would not be something that we could resolve with short read assemblies. And so we would just kind of move on not knowing that that was there. But at the same time, now we can technologically cover those gaps with long reads. We can polish this with short reads, get these high quality assemblies, but it doesn't reflect what's actually going on. And here's one more example, and this is with Will Shropshire, who is a PhD student of mine that graduated a few years ago. He's now over at MD Anderson doing a postdoc. And this is another example of how these things can be inducible. And so we have these longitudinally collected E. coli isolates that developed a non-carbapenemase producing carbapenem resistant phenotype. So essentially, we don't have the gene that degrades the carbapenem, the antibiotic, but it still has that same phenotype. You can't treat this bacteria with that specific antibiotic. And what was interesting is that this appeared to be inducible. So what we saw was that early on, and particularly on the instruments that are used in the clinical lab, this looked to be sensitive to this antibiotic. But then as you would grow it in that antibiotic, it would look to become resistant very, very quickly. And we actually were able to show experimentally, reproducibly, that this organism becomes resistant in 24 hours every time you subject it to the antibiotic. But essentially what we found here is we had this large mobile genetic element, and we're going to call it a transposable unit because it doesn't actually fit the definition of a transposon or an integron. It's just kind of weird. It's like a Russian nesting doll of different things. But essentially what we found is that as you would go through, you have the index isolate here that was sensitive to the antibiotics. After about 2 days, we have an isolate from that same patient. Now we have 10-fold coverage of just this little region of this transposon. And when we went and looked at the context of where this was in the chromosome, we saw that roughly there were 10 copies. We know they were all inserted in the same location, so we ended up having 10 tandem repeats. But some of these had 3 copies, some of these had 15 copies. It wasn't something that everybody had 10. This was not locked in. There was variability here. On the third sample that we collected the next day, this is where it became that carbapenem-resistant phenotype. We actually noted that roughly 3x of this jumped into a porin, knocked out the porin, and that's what causes this phenotypic change. And then a couple more days go on, and it jumped into an unknown context, which is just in a hypothetical gene. We don't really know what that did yet, or does yet, but at the same time, this was something that was reproducibly able to happen. And so we see that in this situation, we know the context of the insertions, which biologically was more important for us, but that they're not all exactly the same number of repeats. They're not all exactly the same structure because each one of them has a different mechanism or a different application basically of this tandem repeat. It is something that is occurring in multiple cells, not just once and then a selective advantage. So we don't quite reflect this without getting way into the weeds of individual reads and using single molecule-based analyses to be able to understand this. And just to show that this is not a new concept, this is a paper from 2004 showing a number of different mechanisms that bacteria used to do things like phase and antigenic shift. Some of them were structural, some of them were enzyme-based, and showing that we've known about this for a very, very long time. It is only just recently that we've been able to really get at this level of heterogeneity within our genomic data accurately. So why do we care? What does it have to do with assemblies, right? So our assemblies here, as we go through, we have our DNA extraction, and this is really what we're trying to recreate, is this structure of DNA. The whole point of doing all of this work is to informatically reflect what is happening biologically, whether that's within a pathogen in a host-pathogen interaction sense, whatever that may be. And so we take this DNA, we fragment it down into these small fragments, and even Oxford nanopore, we're still fragmenting it down, whether that's with ligation or a transposase-based kit, where the transposome comes and clips the DNA. And we're always fragmenting it down into some piece, just that the value of the long reads allows it to fragment it into a much larger fragment. We go through, we ligate on our adapters, we sequence it, and then we assemble that sequence. We take all of our little pieces, figure out the order that they go, and we try to create our consensus genome. Hopefully, in that situation, this consensus is complete, and we're able to go and look at this from a linear structure of a circular biological structure. And this is the goal. But if we have some sort of heterogeneity in the data, right, and we align it up here, the short read assemblies just toss that. It fractures at that point. That's why you have multiple contigs. It's usually the place where those contigs stop is where there is some sort of heterogeneity or something that you can't cross with the length of that read that is similar to in multiple locations in the genome. If we go through the long reads, we can actually align them and say, okay, they're in the same context, but now we have these two different answers. What do we do with this? How do we judge and how do we make the choice? And many of these things are based on heuristic algorithms, some sort of underlying machine learning method, some sort of clustering that goes through and makes the choice, but it becomes very, very difficult to represent that. So how do we do that? What does a consensus mean when heterogeneity is present? I don't have a clue. I'm not sure yet. This is really the front end of this, and I hope you see why this is important, because I'm hoping that my group is working on this. I'm going to try to convince Todd to come help me with this problem, but I think it becomes incredibly impactful, and it's not something that just one group can solve. And with this many people who are all very computationally savvy, interested in this, I think that nexus of biology and computation is where we have to be to answer these questions. And so I'm hoping you take this as a little bit of a call to arms to be able to start to work on this. And what it becomes then is a situation where we are trying to differentiate technological artifacts from biological variation, which is basically just a different way of saying how do we know something is real. And so this is a paper here, and I will say that this paper is published by the maker of Unicycler. Very strong science, very strong group, they do great work. Never take someone's word when they tell you that their tool is the best, though. Validate your stuff yourself, check it yourself, and what this is showing is that you have a number of different assemblers for long-read data, and they're showing that there is a bias away from very small plasmids in many of these assemblers, and some of them miss stuff on the larger size too, but they're using Unicycler as their gold standard. They don't check to see if these plasmids actually exist, if they're nested in each other, if there's heterogeneity, what's going on here. And again, that's not a criticism so much as it is something that we just need to be aware of. When you publish a tool, you're benchmarking it against other people's tools. It is in your best interest to make it look good, right? We also often test these on reference sets that don't reflect the more challenging clinical isolates that we run into. And so make sure you perform your own benchmarks. We do this all the time in the lab. It adds a ton of time to our work, but it also makes us highly confident in our results. So you need to go through and check. And there's many, many pieces of this. So in my lab, all of my mentees go through the full process at least once. And I mean they extract their own DNA, they prep their library, they sequence, they go through the full process of doing the analysis to the final answer on a project at least once. And that's because even things that we would think are trivial, like library preparation, really, really matter. And so here, you can see that we have kind of two different methods that we often see, particularly within things like Oxford nanopore sequencing. So we have sheared DNA and unsheared DNA. And so our sheared DNA methods are what are required for things like ligation kits, where you have to go through and do some sort of shearing, because if you're going to ligate adapters onto a piece of DNA, inherently it has to be linear. There has to be pieces at the end for you to ligate that DNA onto. If you have a 3 kilobase plasmid and you didn't do any shearing, there's nothing for you to ligate onto. And you can actually see that, and I find this very entertaining. This is one of the articles from Oxford Nanopore that shows here on this side, where you go through the ligation prep, you have this kind of shearing, but you've missed these 2 small plasmids. You can't pick those up with a ligation-based kit, and it becomes very impactful. When I was preparing for this, I was reading through a number of these kind of online tutorials and papers, and there was one that was benchmarking a tool that said, well, we saw a low copy number plasmid, but because it was low copy number, we just threw it out. They used ligation kit. It's not low copy number, it's there. They thought that was an artifact, so they removed it, but it's because of the method that was chosen that they found what they found. On the other side, if you go and you use the transposase methods, things where you would actually go through like the rapid kit for Oxford Nanopore, now you can see that transposome complex goes through and nicks those little plasmids. And so we see this in our data all the time. We will have complete circular plasmids that were nicked once, and we can get single molecule, single structure based on that kind of data. But even small decisions like this matter. And when you go through Oxford Nanopore's webpage, and they tell you how to sequence the perfect bacterial genome, they tell you to use the ligation kit. Depends on what your goal is, right? Depends on what you're trying to achieve. And that's also another situation where different assemblers matter substantially. And so see Shelby is over here grimacing. This is one of her isolates that is giving us fits at the moment, because we're going through, and this is with this is with Fly. And Fly is considered the current gold standard for long read assembly. It goes through and it bins reads based on k-mers, based on prevalence, and a number of other ideas. And the idea there is that if you have a high copy number plasmid, it should be more prevalent than, say, the chromosome is, so you're able to bin based on those different levels. It's also a fundamental underlying technique for things like metagenomic assembly methods, where you bin based on k-mer prevalence and other mechanisms to try to differentiate what goes together. That said, the binning of the k-mer obviously doesn't work completely in this specific example. And so when we go through, Fly tells us this is circular, but when you start to look at it, you do see that there are reads on the left and reads on the right that are pairs. So yeah, there are things at the end that wrap around to the other end, which is what Fly uses to define circular. But we also have whatever this booger is in the middle, and there's actually no coverage here. And that's because what we're seeing here and what we think we're seeing are 2 very similar plasmids that Fly thinks are the same because they are within a certain nucleotide difference of each other. And it's just throwing the place where they don't overlap in the middle and saying, it's circular. But you can see that on this side, all of these different pieces, you actually have reproducible patterns where single reads match one specific structure and other reads match another structure. And this consensus represents 2 biological states. How to disentangle this, we're still working on. I think we can take this and further bin these reads, pull them apart and redo microassemblies of the specific structures to be able to pull them apart. We're also looking at single molecules to see if we can span these gaps. But doing this in an automated fashion is really where we're at now, at the very front of how far we've gotten. And so we don't have all of these tools ready. I would love to say, hey, we've got this package, please use it. We're not there yet. And that's not the only situation that goes through. So we also have issues with defining things like circular. So again, this is another one that Fly says is circular. This is a linear plasmid. This is biologically never going to be circular because it does not exist in that state. But when you go through, because Fly picks an arbitrary location to set the ends of that representation of the genome, this piece in the middle is actually where it should be ending, but because again there are reads that map to both sides of that junction, it thinks this is circular, whereas this is supposed to be the linearization point. It doesn't perfectly reconstruct this. We're still working on some of these methods. But again, this is something that we would only know because we're in the weeds looking for this, and only because we're really checking, and by we I mean Shelby. But essentially, these are situations where we're really getting into inventorying all the ways that the biological heterogeneity, these phase shifts, these structural variants impact the way that we think about what a consensus is. And right now, this is okay because we're representing gene synteny. We're looking at understanding what genes are there and what are absent. But as we get into more complex assessments of how these things interact with the host, how they are becoming more recalcitrant and form antibiotic resistance in a population, understanding that heterogeneity, understanding how some of these different colonies are sequestering antibiotics and acting as a bait for the rest of the colony so that they can all continue to persist, becomes incredibly impactful. And so right now, we're sitting with existing tools that either run a single assembly, which can reveal structure, sometimes gets it right, but sometimes can't reflect this heterogeneity and they will stitle break, similar to our short read assemblers. Or you end up with things like these consensus of multiple assemblies, which is what tricycler is that I call out here. And these are looking to be nice tools because it's essentially bootstrapping of your genome assembly. But again, it's a consensus. And so as you go through, if you have a consistent bias, common bias based on your assembly method, if you picked the wrong library prep kit and you've missed a plasmid, you're still not going to pick it up, no matter how you specifically go through and do this. And so we need better full-length ways of going through this. And so this is my call to action. This is where I want you all, hopefully, if you're interested in this, to start to jump in and really help solve these issues. This is not a condemnation of current technologies and techniques, right? We are always building and moving forward, and once we find this next step, there will always be something else that is not quite done right? We need to keep moving forward, but we need to understand the limitations of these current tools, and we need to know that our consensus assemblies do not always reflect biological variability. Don't take any informatics tool as ground truth, particularly too as we move into things like machine learning and AI where we have black box tools. It's very hard to understand exactly what they're doing unless you benchmark them against a wide range of different situations. And we also need new tools to assess how these different assemblies come together, differentiate these technological artifacts that I've been describing from the biological variation. And I honestly think, when you look at the Human Genome Project and you look at human genomics and cancer genomics, they've moved past the FASTA assembly. That is not the unit of analysis anymore. In bacterial genomics, that's still what we use. And all that can represent is a single string of DNA with no indication of variation, no ability to layer in more data. I really think that we need to move past that and move into better ways of modeling this level of heterogeneity and variability. But it all comes into understanding why and being thoughtful about how we do this. So do I have all the answers? Absolutely not. Are we trying to inventory the challenges so that hopefully myself and people that are smarter than me can start to jump in on this? Yes. And we need tools that start to reflect this heterogeneity because it's been proven to be important. Not only just bacteria, but low-frequency variants in cancer genomics are incredibly impactful. We see this utility all over. Now is the time to continue to move past this and really start to move into a new phase of how we think about this. And I think the path is there. We just need people to be working on it. And so that's what I want to leave that for today. Hopefully you've gotten something useful out of this. But I'll take any questions. Thank you.
Questions? Blake?
Got one in the middle.
That was a great talk. Quick question. You mentioned tricycler, and I've been struggling with tricycler.
Sure.
So I think the idea is to have 6 different assemblers put together and randomize your samples by subsampling, and then. But have you made it work? Because mine is not working.
So we have, but it doesn't always give us the right answer. So it does And so actually I was looking at this over the weekend because the original paper did tell you to use multiple different assemblers and run multiple assemblers to get over assembler-specific biases. But Ryan's most recent kind of use cases and going through that is actually now he splits it into 3 sets of data and then runs them, they're all just random subsets, and he runs them all through Fly, and then he checks them against each other. And so if you have issues, so like my complaint about Fly is that high copy number plasmids almost exclusively get kicked out. It should not be doing this, it's a known thing, but you have a huge false negative rate with small plasmids, which was shown in that graph that it showed. Tricycler doesn't fix that because it's a systematic problem with Fly. And so I really like the idea of Tricycler from a fundamental point of kind of bootstrapping your assemblies, but it still has the same issues of whatever you're basing it off of. We have gotten it to work, but it's not an easy tool to work with. Yeah. Yeah.
Thank you for the great talk. I have a question regarding the long reads specifically. Yes. We have 2 main companies, Oxford Nanopore and PacBio. Did you see any shift in accuracy, precision specifically, using Oxford Nanopore versus PacBio or the other way around?
Yeah.
Additionally, if mixing the 2 technologies will add something new to the assembly, or it will be non-economically efficient?
So it's a fantastic question. So I'll break it down into 2 pieces. So there's one situation, so Oxford Nanopore gives you much longer fragments than PacBio ever will. We don't tend to work with PacBio much because there's actually a length versus accuracy handoff. Even when you're using the consensus reads, if you go with 40 kb or 60 kb library preparation methods, you are inherently going to then have less times around the circle, and you're going to have less accurate data. And so you have a length versus accuracy trade-off there that most of the assemblers don't take into consideration. We've also found that most of the PacBio methods, because they use things like the blue pippin or the sage elf for size fractionation, miss all of the little plasmids as well. And so this is another situation of the library prep that we've run into. If you're trying to go for less systematic bias, PacBio tends to have less issues with kind of consistent problems with methylation because there's amplification steps and different pieces in there. So it's a bit easier to work with that in some ways. But I like the structure of the longer reads and longer fragments much better. I still don't think you can get away from having short read polishing. And so if I'm already gonna have to do short read polishing, then the long reads to me are more about structure and being able to cross these large pieces and represent the entire genome. And so I'm gonna take the longer pieces, even with a higher error rate, to be able to work with because it's also consistent rather than the PacBio pieces. And so I think that both are viable options. This is just the way that my lab has gone. It's also way cheaper to have an ONT device than it is to have a PacBio device. And when you're trying to subsidize all these subcontracts to be able to keep the machines on, that does make a difference. But I think they're both viable. It's just that I think a lot of the bacterial world has gone to ONT because of how cheap it is to get them and run them, and then you get these long fragments. And we can get megabase long fragments. Can't do that on PacBio.
Another question on the same topic. We have some species that really, really close to each other, homologous. From my understanding, PacBio have an error rate, let's say around 3% more or less. I will leave it open. PacBio is a little bit more accurate. Mm-hmm. Focusing on Oxford Nanopore like you are using, are you able to differentiate between these highly homologous or similar species, or it will be a problem with the error rate? So
the way that we run it, and we've actually got some publications out there, we have a pipeline that we put together that we're currently revisiting that's all hybrid based. So we do structural assemblies using Oxford Nanopore data. We'll polish that with Oxford Nanopore data, but that only still gets you to about 99% accuracy. Then we actually go through and do short read polishing with Illumina data, go through and do error correction and repeat sections, and we've come through and on every one of these kind of that we've checked for, ignoring the heterogeneity problem still, we're talking 99.9999% accuracy. We're in the same level as the RefSeq type stuff. So we can even take, and this is actually Shelby's project, longitudinal isolates from the same patient and differentiate SNPs across time very accurately, and then look at how that changes recalcitrance. What are they doing to be able to avoid the immune system and antibiotics and things like that? And so we're at the point where we're able to differentiate very small differences, both structurally and from a nucleotide point of view, because of that combination of data. And I think it works really well for closely related species, closely related strains. It requires a lot of hand checking, but it is something that I feel very comfortable with and confident in the results that we can get from that.
All right. I got a couple questions. This is fun. Any other questions for Blake? It's okay. We're a little bit over time, but it's good. We have, again, buffer built in here. Reminder for the questions for the panel, I submit them soon because we're going to get started on that. But yeah, I guess, Blake, I guess we could talk here probably for an hour if
A
we
long
wanted. Comment. So again, coming back to an earlier talk that we saw from the keynote, there's some interesting observations. One, I would like to, again, on Team Microbe here, say, you showed some pretty complex regions and repeats that absolutely required long reads. I think a common misconception I would get is bacterial are simple to resolve, right?
Yeah.
So why do you even need long reads? I think you presented a good case for that, but you often feel that just out of your experience underappreciated, like the challenges in this? Because I think we can learn some stuff from what, in the human genomics community, how they're actually deconvoluting complex structural variation with long reads.
Yep.
Have a lot of momentum thanks to standards. I think Fritz has a paper recently, it's called "If You Can't Measure It, You Can't Improve It." Like, do you feel like we're just behind them? But what do you think the state of the field is?
Yeah, I think that because the Human Genome Project was such a big push, the human genome momentum was much earlier. I mean, you saw the Human Microbiome Project come along, and that, well, a big part of the first wave of that was to just generate reference genomes for a lot of these different organisms that were coming out of the microbiome. But even that, was a ton of work. And so I think a lot of people think that bacteria should be easy because they're tiny, but we don't have references. We don't have a human genome to be able to go and look at that's been heavily curated that we understand. And structurally, they are incredibly diverse. And so I do think that because of that, I mean, in the tools and the technology, microbiology is inherently a slow field to adopt new things like this. They still, if you mutate an organism, you just confirm the specific region that you mutated with Sanger sequencing. And if you try to say, I actually shotgun sequenced the whole genome, and look, I have no off-target mutations, they say, oh, we still want you to confirm with Sanger. And so I think we're just behind in a lot of ways. And I think also, so there's a really cool book called Zoobiquity, which actually looks at this idea of how veterinarians have actually been answering a lot of these questions in zoo animals that now we're starting to realize we see in humans as well. And so heartbreak, and I forget the name, there's a syndrome, but essentially, like, they've been observing this in monkeys in zoos for 100 years. And it took until the 2000s for people to say, oh, we actually see this in people, and here's the name of these two different things, and we actually know more about it in animals, because there's this idea in humanity that we are different, right? Even though we are apes, mammals, we're all the same thing. There's different layers of this. We like to treat all of our own conditions differently, and I think the bacterial world is similar. We don't tend to learn from cancer genomics, from human genomics, as much as we should, because inherently we don't have introns and exons, and so people go, oh, we have to have totally different methods. And I don't think that's real. I think that you're right. We need to have better crosstalk, and the standards in the human genetic space need to come over.
Yep, and then just taking organizer privilege of asking more questions here. Going back to some of the things that you talked about for PacBio versus Nanopore, and again, maybe we can talk more about this on the panel, but something struck me that even I've spoken to this about, with Fritz before and even MedHat, but the pros and cons, if you're saying that it's throwing out plasmids due to their length, in some sense it suggests to me you can never use this for wastewater then, because everything's already degraded. Like, is PacBio viable for environments that we expect degraded, sample, and we don't even know the size? So even things that should be longer length are gonna end up being shorter length. And because a common question I get is, why are you not using long read sequencing in these environments to really tease things apart? And I just come back to them saying, look, it's not as simple as that, right?
Yeah, I think you can, as long as you're cautious about the way that you do the generation. But again, similar to Oxford Nanopore telling you to use ligation kit, when you look at PacBio's guidance on bacterial genomes, they tell you to size fractionate. And they want you to use like the G-tubes to be able to do the shearing, and then you ligate on the smart bells, and then you run it through a Sagel for a blue pipette to size select, and then you have this high quality library that you can get the longest fragments as possible. And so if you're going to go for wastewater, I think it's a totally applicable way to do so, but you can't do size fractionation, and you have to be okay with small average insert sizes, things that would normally throw flags in PacBio's software suite. You just have to be like, yeah, that's actually what I want, because it is degraded, because it is different size fractions. And so I think it just takes an understanding of the whole process and selecting the library preparation, the fractionation, all those different pieces intentfully for the question that you're trying to answer, which is not what we often see in the guidance. And so that's why my lab does everything once, at least, is because I want them to see how the decisions that they make in the wet lab, where people often think, oh, that's just standard, that's been solved for years. It hasn't. There is still nuance and art to it that you have to be thoughtful about, and I think the same thing would be true of PacBio. You can totally use it, you just have to be thoughtful about how you prep it.
Great. Let's thanks Blake, but all the speakers from the first session today, just thank them all.